For decades, a spinal cord injury has meant a life sentence of paralysis. The intricate wiring of our central nervous system, once broken, has been considered irreparable, a biological rule with no exceptions. The human body is a marvel of self-healing, but the spinal cord has always been the tragic outlier. But what if we could defy that rule? What if we could actively rebuild what was lost, printing a living bridge to span the site of an injury and coax the body to repair itself? This isn't science fiction anymore. At the intersection of neuroscience, engineering, and biology, a revolution is underway. Today, we're exploring the audacious science of 3D printed scaffolds, a technology that offers a tangible blueprint for repairing the irreparable. To understand just how revolutionary these scaffolds are, we first have to appreciate the sheer biological hostility of a spinal cord injury. It's not just a simple break. When a traumatic injury occurs, the initial impact kills neurons and severs the long axonal tracts that act as communication highways between the brain and body. But that's just the beginning. The body's emergency response, called the secondary injury cascade, makes things much worse. Blood flow is cut off, causing more cells to starve and die. Dying neurons release a flood of toxic chemicals that overexcite and kill their neighbors. The immune system rushes in, causing massive inflammation that adds to the chaos. In the days and weeks that follow, the injury site transforms into a war zone, completely inhospitable to any attempt at natural healing. Two major barriers form. First, a dense, interwoven network of cells, called glial scar walls, off the injury. While this is a natural attempt to contain the damage, this scar tissue also pumps out a cocktail of chemical signals that actively repel any new nerve fibers called axons that try to grow across. It's like putting up a chemical force field that says "do not cross." Second, the damaged tissue often breaks down and liquefies, forming a fluid-filled hole known as a cystic cavity. This creates a physical gap, an empty chasm that regenerating axons simply cannot traverse. The very architecture of the spinal cord is gone. This multi-layered, hostile environment is why natural repair is impossible, and why a simple drug or therapy isn't enough. You need to solve multiple problems at once. You need a structure. This is where neural tissue engineering comes in, with the biomaterial scaffold at its core. The concept is brilliantly straightforward. If there's a gap, let's build a bridge. But this is no ordinary bridge. Scientists are designing these scaffolds to perform several critical jobs simultaneously. First, and most obviously, they provide structural support. They physically span the cystic cavity, giving cells a surface to latch onto and axons a path to follow. Second, they provide axonal guidance. By engineering the scaffold with specific features, like microscopic parallel channels, they can act like highways, guiding the regenerating nerve fibers in a straight, organized line, mimicking the natural structure of a healthy spinal cord. Without this guidance, new axons would grow randomly, like a tangled mess of wires, failing to make meaningful connections. Third, the scaffold can act as a physical barrier to inhibit the formation of that inhibitory glial scar. By occupying the space, it prevents scar-forming cells from infiltrating and creating their chemical wall. And fourth, it acts as a sophisticated delivery vehicle. Scaffolds can be loaded up with a payload of therapeutic agents, living cells, powerful growth factors, and other helpful molecules, and release them in a controlled, sustained way, right where they're needed most. To do all this, the ideal scaffold must have a specific set of properties. It must be biocompatible, so the body doesn't reject it. It needs to be biodegradable, gradually dissolving over months, as new, healthy tissue takes its place, avoiding the need for a second surgery to remove it. Its mechanical properties are also vital. It has to be strong enough to provide support, but also soft and flexible enough to match the delicate spinal cord tissue, so it doesn't cause more damage. Early methods for making scaffolds created porous structures, but they offered little control over the internal architecture. This all changed with the advent of 3D printing. or more specifically, 3D bioprinting. This technology allows scientists to build complex, patient-specific scaffolds layer by layer with unprecedented precision. Using an MRI scan of a patient's injury, a scaffold can be designed to fit the unique geometry of their specific lesion perfectly. Several different 3D printing techniques are being used. The most common is extrusion-based bioprinting. Think of it like a very precise robotic pastry bag, extruding a continuous filament of a gel-like biomaterial or bioink to build a structure. It's versatile and can print with high densities of living cells embedded right in the ink. Another method is inkjet bioprinting, which works much like your desktop printer. It deposits picoliter-sized droplets of bioink, and it's generally faster and gentler on the cells. For ultimate precision, there is laser-assisted bioprinting. Here, a focused laser pulse propels a tiny droplet of bioink onto a substrate. It's a nozzle-free technique that can print single cells with incredible accuracy and boasts a cell survival rate of over 95%. Finally, there's stereolithography, or SLA. This technique uses a projected pattern of light to selectively harden a photosensitive liquid resin, layer by layer. SLA is fantastic for creating incredibly intricate microarchitectures, with resolutions down to just a few microns. A new version of this, called microscale continuous projection printing, is mind-bogglingly fast. It can print a 2mm implant, perfect for a rat's spinal cord, in just 1.6 seconds. The true power of this technology is the ability to create scaffolds with a biomimetic architecture, meaning it mimics biology to actively guide regeneration. A key design principle is contact guidance. Scaffolds are often printed with dozens of parallel microchannels, each about 150 to 200 micrometers in diameter. These channels act as those highways we talked about, preventing growing axons from getting lost and encouraging them to bridge the lesion. In studies with rats, scientists have watched as axons from the host's own spinal cord grow into these channels, aligning in parallel arrays, just like in a healthy spinal cord. Some designs even mimic the distinct gray and white matter regions, printing a solid core to represent the neuron-rich gray matter and a channeled outer region to guide the long communication tracks of the white matter. The material used to print the scaffold, the bioink, is just as critical as its design. Most bioinks are hydrogels, which are water-swollen polymer networks that have a soft, squishy consistency, similar to our own tissues. There's a whole library of materials to choose from. Natural polymers are often derived from our own bodies, making them highly biocompatible. This includes collagen, the main structural protein in our bodies, hyaluronic acid, a major component of the neural environment, and fibrin, a protein involved in wound healing. On the other hand, synthetic polymers like PEG, PEG, and PCl offer the advantage of highly tunable properties. Scientists can precisely control their strength and how quickly they biodegrade. Most often, the best approach is to create composite bioinks, blending natural and synthetic materials to get the best of both worlds, a scaffold that is both biologically active and structurally robust. But a scaffold alone is often just an empty house. To truly kickstart regeneration, you need to furnish it with living components. This is where cell therapy comes in. Scaffolds are frequently loaded with a payload of living cells to replace those lost to injury and to provide support for the surviving neurons. The most direct choice is neural stem cells, which have the potential to become new neurons, astrocytes, and other essential nervous system cells. When loaded into a scaffold and implanted, they can integrate with the host tissue, extend new axons, and form neural relays that bridge the injury gap. Another powerful cell type is the mesenchymal stem cell, or MSC. Harvested from bone marrow or fat tissue, MSCs aren't great at becoming neurons themselves, but they are incredible little factories. They pump out a huge range of neurotrophic factors and anti-inflammatory molecules that help create a much more pro-regenerative environment. Perhaps the most exciting source is induced pluripotent stem cells, or iPSCs. These are adult cells, like skin cells, that have been reprogrammed back to an embryonic-like state. This allows scientists to create patient-specific neural cells, avoiding immune rejection. These iPSCs can be turned into neural stem cells and then loaded into the custom-printed scaffold. To enhance this process even further, scaffolds are infused with bioactive molecules. These are biochemical cues that tell the cells what to do. We're talking about neurotrophic factors, proteins like BDNF and NT3, that act like fertilizer for neurons, supporting their survival and growth. Scaffolds can be designed to release these factors in a sustained, controlled manner, sometimes even in a gradient, to better attract axons in a specific direction. They can also be loaded with enzymes, like one called chondroitinase ABC, which has the specific job of chewing up the inhibitory molecules in the glial scar, effectively clearing a path for regeneration. The field isn't standing still. The next wave of innovation is already here. Researchers are now exploring conductive scaffolds. The nervous system runs on electricity, so by incorporating conductive materials like graphene or carbon nanotubes into the bioink, it might be possible to restore electrical signaling across the injury site. These conductive scaffolds have been shown to enhance neural differentiation and axon growth, acting as a better platform for transmitting the body's natural electrical signals. Then there's 4D bioprinting. This adds the fourth dimension, time, to the process. 4D printed scaffolds are made from smart materials designed to change their shape or properties over time in response to stimuli in the body. Imagine a scaffold that is initially rigid for implantation, but then softens over a few weeks to perfectly match the surrounding tissue. Or one that changes its shape to guide different stages of the healing process. While the results in animal models have been incredible, the path to using these scaffolds in human patients is a long one. In numerous studies, rats with severe spinal cord injuries who received these 3D printed, cell-laden scaffolds have shown remarkable recovery. We've seen significant axonal regeneration across the injury, a reduction in scarring, and most importantly, measurable improvements in locomotor function, including the recovery of hind limb movement. In some cases, treated rats went from being completely paralyzed to being able to walk again, albeit with a limp. These are astounding preclinical successes, but moving to humans presents huge hurdles. One of the biggest is vascularization. Any implanted tissue needs a blood supply to survive. Researchers are tackling this by designing scaffolds with pores that encourage host blood vessels to grow in, or by preloading the scaffold with the types of cells that form blood vessels. Safety is also paramount. The implants must be proven to be non-toxic, and when using stem cells, the risk of tumor formation must be eliminated. Clinical trials are in their very early stages, primarily testing the safety and feasibility of implanting simpler scaffolds in human patients. The results so far have been cautiously optimistic, showing safety and some hints of functional improvement. This convergence of neuroscience, material science, stem cell biology, and advanced manufacturing is forging a new path for what was once untreatable. 3D bioprinting provides an unprecedented ability to design and fabricate living implants that are active participants in the healing process. The journey is far from over, but the progress is undeniable. The ability to print a custom blueprint for repair, a living bridge loaded with the tools for regeneration, represents a profound shift in medicine. The vision of reversing paralysis is not yet a reality, but for the first time, the science is no longer fiction. The printers are humming with the promise of a future where spinal cord injury is no longer a life sentence.